Good evening, everyone. I welcome you all to the Week 7 live problem solving session on NPTEL Bioinformatics Algorithms Applications course. So, in this course, we'll be seeing about computational protein structure analysis, followed by introduction to the protein stability. Computational protein structure analysis is a broad field that involves various techniques and tools for studying the structure, function, and dynamics of the proteins. So here are some of the features of computational protein structure analysis, that is structure prediction. One of the primary features of this computational protein structure analysis is predicting the 3D structure of a protein from its amino acid sequence. This involves using algorithms and computational methods to predict the most energetically favorable conformation of the structure of the protein. Next is structural alignment. Structural alignment is the process of comparing 3D structures of two or more proteins to identify similarities and differences. It is useful for understanding evolutionary relationships, identifying functionally important regions, and designing new protein structures. Then we have molecular docking, which is a computational technique used to predict the binding mode of a ligand or a drug molecule or a small molecule to a protein. It involves generating different conformation of the ligand and protein and then calculating the energy of their interaction. Then we have molecular dynamic simulations. So these are used to study the motion and behavior of proteins at the atomic level over time. So these molecular dynamic simulations involve solving Newton's equations of motion for all atoms in the protein, uh, researchers which allows researchers to study protein dynamics, conformational changes, and protein ligand interactions. So here I have a question tells which of the following is the primary feature for protein structure analysis? So the answers can be provided using the chat box. So which of the following given options is the primary feature or one of the features of protein computational protein? So the only option that deals with the structure analysis is the structure prediction. So as we have discussed, one of the primary features of computational protein structure analysis is predicting the 3D structure of a protein from its amino acid sequence using several algorithms and computational methods, wherein in this we try to find out the most energetically favorable native conformation structure of the protein. So amino acid composition is again a sequence-based features. BLAST, sub basic local search alignment search tool. It's again a sequence-based similarity or homologous sequence finding tool. So the answer is protein structure prediction. So in the last class we have a demo to PDB param, but still PDB param is here again. So to discuss some of the features that we didn't do in the last class. So it has a comprehensive set of structure-based features derived from protein three-dimensional structure. So this is a web server that provides parameters based on protein three-dimensional structures. So here in PDB param, the features are grouped into four main categories. That is physiochemical properties, interresidue interactions, secondary structure propensities, and identification of binding sites in protein complexes. So the first one is physiochemical properties. These properties describe various characteristics of protein structures related to their chemical and physical properties. So they include parameters such as center of mass, surrounding hydrophobicity, paridness, and non-covalent interactions. Then we have interresidue interactions. So wherein these parameters focus on understanding the interactions between amino acid residues, the protein structure. So interresidue interactions include short, medium, and long range interactions or contact order, long range order, total contact distance, Then we have propensities. So propensities are the parameters which assesses the likelihood of amino acid residues to adopt specific secondary structures such as alpha helix, beta strands, or turns, or wherein in other ways it can be turned as a given a stretch of sequences. These propensity values could help in predicting whether the given sequence could acquire the fold of a alpha helix or a beta sheet. So this is the principle behind the Chow-Fasman method that we have discussed in the secondary structure prediction methods. Finally, we have binding site. So this is a protein complex related feature which helps in locating residues involved in protein DNA or protein RNA. So briefly, it is protein nucleic acids or protein ligand. So here, the identification of binding sites is based on distance criteria. So with this, I want to conclude about PDB param, telling it offers a valuable resource for analyzing protein structures and understanding their functional implications by providing access to a wide range of structure-based parameters. So let's take a task on PDB param. So using PDB param web server, report the center of mass of the protein having PDB ID 2TRX and chain ID of A. So for this question, you can enter into the website PDB param, then click on compute button, then fill in the required input details, then select the physiochemical properties, then press submit. Under the physiochemical properties, we have subset of properties, of which select the desired one. Here it will be center of mass. Then click on submit. You will be redirected to a results page. Yeah. No need to mention the values. If you are just done, you can just type in the chat box that you have completed the task. Then we'll proceed one more task on PDB param. So first, to this, visit the PDB param. So in the Google, we can directly type PDB param. Once you enter into the home page of PDB param, click on compute button. So now you will be redirected to an input page. So in the input page, fill all the required details. The details are nothing but PDB ID, which is 2TRX, and chain ID A. Then under input option, select physiochemical properties. There is a radio button given. Then click on submit. So under the physiochemical properties, PDB param has set of other properties, under which select center of mass, then click submit. Okay, let's move. So this is the home page of PDB param. So I'm moving on to the compute. So under the compute, we can see please enter the following. So what we need to enter is PDB ID and chain name. So I'm entering the PDB ID 2TRX and chain name A. And now I'm selecting physiochemical properties and then selecting submit. So why these selections? Because there are set of features that are grouped into four main categories, that is physiochemical, interresidue, secondary structure, and identification of binding site. So center of mass is grouped under physiochemical properties. So I am selecting a physiochemical properties here. So when now we go into the feature selection tab. So here center of mass is present. So I'm just clicking on center of mass. You can see the blue tick appeared. Then I'm clicking on submit at the bottom. Submit. Yeah, here is the result. So the result page shows center of mass of PDB ID 2TRX.PDB of chain A. First it gives molecular weight, which is 13.305 kilodaltons, and followed by center of mass. Center of mass has three coordinates. It is again a representation of a grouped Cartesian coordinate system. So the x value is 32.954, y value is 31.669, and z value is minus 0.745. This is center of mass of the whole protein 2T-Rx. This is what the results we got over here. Okay, similarly, now we'll try to identify some non-covalent interactions in PDB param. So here I have given the same PDB ID and chain ID, and you can log in into the PDB param and then try to report here number of disulfide bridges, number of ionic interactions, number of hydrophobic interactions, and number of aromatic interactions. So PDP param provides these structural parameters wherein in field of computational biology, where most of the people deals with the machine learning models and all. So machine learning requires a quantitative input. So these parameters acts as an input, quantitative input to our problem. So wherein the machine learning models can be built and future prediction can be made. Thank you for your response. Okay, I'll show a demo here again. So similarly, in the physiochemical properties page, we can see disulfide interactions, ionic interactions, hydrophobic interactions, and aromatic interactions in the first line. Once after selecting these, I am clicking on submit. So the list of interactions along with their residue name, chain name, and distance between them has been given in a tabular format. So in disulfide bridges, we have only one disulfide bridge, that is between cysteine 32 and cysteine 35. And under ionic interactions, we have set of ionic interactions. So we can count. So 1, 2. So we have nearly 20 ionic interactions and around 15 to 16 hydrophobic interactions, followed by 4 aromatic interactions. This is how non-covalent interactions, number of non-covalent interactions that were present in a protein is identified using PDB param. So here is the detailed number of interactions that are present in the PDB ID 2TRX of chain A. Okay. So moving forward, I have one set of couple of questions. So which of the following does not belong to the PDB param server function? So now we have looked into PDB param, what it does. So which of the following doesn't come under PDB param server feature, or which of the following cannot be performed using a PDB param server? So PDB param includes physiochemical properties. PDB param includes interresidual interactions. PDB param includes identification of binding sites in protein complexes. So the one that doesn't PDB param include is molecular dynamics simulation. Okay, moving forward, we have learned about the structure alignment or structure superimposition. So it's a process of aligning two or more protein structures to compare their similarities or differences. Similarly, we have learned protein sequence alignment. Under protein sequence alignment, two concepts we have learned. One is pairwise alignment and one is multiple sequence alignment, wherein we try to identify the conserved regions and evolutionary information from the protein sequences. So similarly, protein structure alignment or protein structure superimposition. So we have three different methods. So first is least square fitting. In this method, imagine you have two protein structures and you want to see how similar they are. So this least square fitting method aligns these structures by finding the best possible match for their C alpha atoms. So this is done by minimizing the overall distance between the corresponding C alpha atoms in both structures, like them overlapping them such that the C alpha atoms fall within the vicinity of other C alpha atoms. That means it tries to make the structures overlap as much as possible with least amount of bending or twisting. So this is how a least square fitting overlapping happens by using least square fit. That is, it tries to reduce the distance between the two C alpha atoms. So while this method is quick and straightforward, but it might miss some finer details of the structures. Then we have dynamic programming. Similarly, we have dynamic programming in sequence alignment. Here we have a dynamic programming in structure alignment. Again, it uses a matrix and compares different parts of the structures, gradually adjusting them to find the best overall alignment. That means it goes part by part alignment. So this dynamic programming is like solving a puzzle where each piece of the structure is moved and rotated until each best fit is achieved. You can think of a puzzle. So we try to fit the puzzle by each part. You take a part, try to fit it. If it is not fitting, try to rotate and check the fit. Still, if it is not fitting, you keep it out and take the new part. That's how this dynamic programming fit works. But this approach is slower and requires more computational approach. Then we have iterative algorithms. So these methods refine the alignment of protein structures to a iterative process. They start with an initial alignment, then repeatedly adjust the alignments or overlaps based on the local structure similarities. If a local structure similarity is found, the alignment is not disturbed for the particular region, and it tries to refine it to the alignment part where there is no proper overlap. So this approach is more accurate than least square fitting and can handle more complex structural differences. Similarly, we have now recent methods like multiscale methods and geometric hashing also. So one such method we'll be discussing later. So I have a question here. What does least square fitting primarily aim to align in protein structures? So in least square method, what does it primarily aim to align in protein structures? Whether it tries to align secondary structure elements or tertiary structure motifs or C alpha atoms or hydrophobic residues. What it considers for the alignment or overlap to happen. So we have discussed least square fitting aligns their structures by finding the best possible match for their. So what it tries to reduce in the overlap. Any answers for this? Okay. Yeah. So least square fitting aligns by finding the best possible match for their C alpha atoms. So it first takes the backbone C alpha atoms and then tries to overlap the two protein structures based on their backbone C alpha atoms. Then this overlap is done such a way that the distance between two C alpha atoms is very minimum. So it considers C alpha atoms for the structure superimposition. So it considers C alpha atoms. So we have a method DALI, so which stands for distance matrix alignment, which is a computational algorithm for comparing and superimposing protein structures. So this is a method which compares structures by aligning their secondary structure elements and assesses the overall topology similarity. So we have seen least square method which overlaps C alpha atoms and then check the overall similarity in the topology. Here this method aligns their secondary structure elements and assesses the overall topology. So the steps in this method include it computes a distance matrix between the pairs of C alpha atoms in the protein structures, then identifies the secondary structure elements and aligns them based on their spatial arrangement. So it aligns the C alpha atoms. So the alignment is optimized by minimizing the RMSD between these aligned SSDs, such that the alignment happens such a way that two secondary structures from these two structures will be close by. Then this method calculates a similarity score by considering the number of aligned residues, RMSD, penalties for gaps and insertions. And finally, it also reports a Z-score to each pairwise comparison, indicating the structural similarity significance based on the uh, similarity score that we have calculated in the previous step. So this method is especially useful in finding proteins with similar folds but with low sequence similarity. Even if there is less sequence similarity between two proteins, we might observe a structural similarity. So this method is useful for finding those, and it is also integrated in various softwares like Dialite program, which is easy to available. In recent studies, this method also has demonstrated the reliability and accuracy in protein structure comparison task, making it a valuable tool in structural biology research. Now, a lot of tools are there, even the RMSD could be fine using PyMol software as well. So with this, let's take a recap of X-ray methods, NMR methods, and cryo-EM methods. So X-ray method, we know it relies on the diffraction pattern produced when X-ray interacts with the ordered array of atoms in a protein crystal. So this X-ray crystallography provides high-resolution structures, capturing detailed atomic positions and bonding interactions. However, this method requires the formation of well-ordered protein crystals, which can be challenging for certain proteins. Additionally, the processing of data collection and structure determination can be time consuming and resource intensive. Similarly, the other method is NMR spectroscopy, so which offers a valuable insight into structure and dynamics of proteins in solution. By analyzing the behavior of atomic nuclei in magnetic field, NMR provides the information on distances between atoms, dihedral angles, interactions within the protein. However, NMR is limited by the size of the protein as signal overlap becomes problematic for larger molecules. Then we have the recent sensation cryo-EM, which has emerged as a versatile technique for visualizing protein structures of macromolecular complexes. This performs by rapidly freezing samples in vitreous ice so that it can preserve their native state and allow imaging under the near physiological conditions. However, cryo-EM can achieve medium to high resolution structures, making it suitable for studying large protein complexes. But cryo-EM requires specialized equipment, expertise for sample and data analysis. So these experimental methods have certain drawbacks or limitations. Experimental methods can be expensive and time consuming, and obtaining high resolution structures through X-ray NMR requires specialized equipment, skilled personnel, and significant resources. Experimental methods often require substantial amount of purified protein sample. This can be challenging, especially for proteins that are difficult to express or purify in large quantities. Experimental methods may not always provide complete structural information, especially for large or flexible proteins. So the regions of the proteins that are disordered or dynamic may be poorly resolved or entirely missing from the structure. So when you observe most of the experimentally derived protein structures and their protein sequence, they have only a particular domain or a particular region of their sequence where structure is solved. So we find missing regions quite often in these structures. So to address these drawbacks, computational methods for protein structure prediction such as homology modeling, ab initio modeling, fold recognition are often employed. So these computational approaches can complement experimental techniques by providing insights into the protein structure and interactions, especially in cases where experimental methods face challenges or limitations. Yeah, so sorry. The overall topology similarity is the overall protein similarity. So by checking the similarity of secondary structure elements, it finally gives the overall topology similarity. Yeah. So starting with this thing, there are several computational methods available for protein 3D structure prediction. So the first one is homology modeling. It is also known as comparative modeling, which is widely used method for protein structure prediction. So it believes that similar sequences will have similar structure. That is, it involves predicting the 3D structure of a protein based on its homology to a known protein structure. This method relies on the assumption that protein structures are more conserved when their sequences are conserved, like similar. So therefore, two proteins with similar sequences are likely to have similar structures. The homology modeling method requires a template structure with high sequence similarity to the target protein and can generate accurate models when template is of high quality. Then we have ab initio methods. These are also known as de novo methods, which predict the protein structure without using any template structure. These methods used physics-based approaches to predict the conformation of a protein by optimizing its energy landscape. Optimizing its energy landscape is nothing but energy minimization. Energy minimization. So these ab initio methods are computationally intensive and requires a large amount of computing resources, but these are useful in cases where proteins with no homology to any known structure is found. Then we have threading. Yeah, the method uses the algorithm to search a library of protein structures for the best match of the target protein sequences. This threading algorithms generates a model by aligning the template structure to the target structure and then refining it using ab initio. And here, these predict the protein structures by matching target sequences to known fold libraries. So they won't match the complete sequence. Here they match a part of a sequence. Then they identify the most compatible fold for the target sequence. Finally, we have machine learning methods. AlphaFold comes under machine learning or deep learning method, where these methods require the usage of large amount of protein structure data to train a model so that it can predict 3D structure of a protein from its amino acids. The advantage of machine learning methods is they can generate accurate models with high speed, but the disadvantage is they require large amount of high quality training data. So each of these methods discussed here has its own advantages and limitations, and they are best suited for different types of proteins and structural features and requirements. Now, to discuss more about homology modeling, the first step in the process is identifying and selecting a appropriate template structure from PDB. PDB is Protein Data Bank, which is a database for uh, all protein structures. So to identify appropriate templates, a pairwise sequence alignment such as BLAST can be done to identify the proteins with high sequence homology to the input sequence. So how can we do this? So we can go into the BLAST P, then input our sequence for which we need to get the structure, then perform BLAST by selecting PDB as the reference database. Then the output will be the list of PDB IDs that are structurally similar, or with a list of PDB IDs along with their similarity to the given input sequence. In these, more sensitive methods are also involved, such as performing multiple sequence alignment or using automated iterative tools such as SciBLAST. So the purpose of BLAST is to find the sequence that is similar to our input sequence and which has a known structure or a resolved structure already. Then in homology modeling, the alignment of query sequence with the template is an important step. It is essential to achieve high quality alignment as it directly impacts the accuracy of the resulting model. A homology modeling can be only good as the sequence alignment. Like a good sequence alignment can give a good homology model. So next we have building a three-dimensional model of the query protein. Sequence alignment is followed by the 3D model building of the target protein. There are different methods used for generating 3D model of the protein based on its templates. So these methods are classified into 4 categories, that is rigid body assembly, segment matching, spatial restraint, and artificial evolution methods. Even when this 3D dimensional modeling building has been done, but there are often gaps or insertions that occur. So these gaps are called as loops or are structurally less conserved during evolution. So loop modeling is an important step in protein structure prediction because loops play a crucial role in determining the function of a protein. However, predicting the structure is a complex process. There are 2 main methods for loop prediction, that is the database search or the conformational search methods. Then we have sidechain modeling. Sidechain modeling is a process of predicting the, now three-dimensional model building is the backbone has been built. For that, now sidechain has to be added. So this process is crucial in evaluating the interactions, and it is usually done by placing sidechains onto the backbone coordinates that are derived from the parent template structure or from ab initio modeling simulations. So this is done by searching every possible conformation of a sidechain. So it is computationally time consuming, and here most of the sidechain prediction programs are use preferred conformations called rotamer or rotamer libraries. So these rotamers are stored in a library which is a collection of preferred sidechain conformations ranked by their frequency of occurrence. So different energy functions and search strategies are used to select the most appropriate rotamer for each amino acid sequence. Then we have model optimization. So model optimization is mostly involves adjusting the position of atoms in the molecule to reduce any clashes or steric hindrance between them, that is energy minimization. So wherein is a process, it calculates potential energy of the model and adjusts the positions of the atoms to minimize this potential energy. Then we have model validation and evaluation. So after the 3D model of the protein is built, it is essential to validate and evaluate its quality to ensure that it is biologically relevant and can be used for further studies. So this involves comparing the model to the real experimental 3D structures and using various evaluation methods to assess the model's stereochemistry, physical properties, knowledge-based parameters, and other criteria. Some commonly used programs for evaluating the protein structures are ProCheck, ProSA. So ProCheck is part of this model that we'll be seeing today. So when these check the quality, it is quietly observed that the quality of the homology model largely depends on accuracy of the sequence alignment and parts of the protein where the template is not similar to the query. So it either depends on template. Then we have ab initio methods. The basic idea behind ab initio protein structure prediction is to generate a large number of 3D conformations of the protein and then to search for the conformation with the lowest energy, which is assumed to be the native structure. The energy of each conformation is calculated using physics-based energy function that takes into account the electrostatic van der Waals and other physical non-covalent interactions between the atoms. The ab initio protein structure prediction involves several steps. First step is to generate a large number of short protein fragments, that is typically 3 to 9 residues in length, that represents the possible building blocks of the protein structure. Next step is to assemble these fragments into a 3D conformation of a protein. This is done by performing a Monte Carlo search through conformational space. So this Monte Carlo search involves randomly selecting and rotating fragments, and selecting is done by evaluating the energy of the resulting conformation. This process is repeated thousands or millions of times to generate a large number of possible conformations. Once a large number of conformations have been generated, their energies are evaluated using the energy function that takes into account all the possible interactions between the atoms. The lowest energy conformation is assumed to be the native state conformation. Conformation is nothing but the structure of the protein. The final step is to refine the predictor structure using energy minimization or molecular dynamic simulations to optimize the structure and remove any steric clashes or structural errors. Some examples of ab initio protein structure prediction method include Rosetta, which is a widely used method and that includes fragment-based approach and physics-based energy function to predict the 3D structure of a protein. Then we have ITASR. ITASR is a hybrid method of protein structure prediction that combines ab initio and homology modeling to predict the 3D structure of a protein. Then we have Quark. Quark is a combination of fragment-based assembly and replica exchange Monte Carlo simulation to predict the 3D structure of a protein. However, predicting the structure of a larger and more complex protein remains a major challenge for ab initio method, but they have made significant progress in recent and been successful in predicting the structures of several small proteins with high accuracy. But still there is a need to improve their accuracy and their reliability. Now it starts with one of the recent method, that is AlphaFold, which is developed by DeepMind. So that has transformed the field of protein structure prediction. Now whenever you access through a UniProt database, that is protein database, you also see the AlphaFold structures integrated to it. When we move into the structure section of any UniProt, you see list of all the PDB IDs, and at the last you see the AlphaFold ID. So So AlphaFold is an artificial intelligence program that is developed by DeepMind that predicts 3D structure of a protein from its amino acid sequence. The program uses a combination of deep neural networks and genetic algorithms to model the protein folding. So today I'll be also demonstrating how input can be given into the AlphaFold collapse file, and then structure can be desired structure can be generated using the file. Its architecture consists of multiple components and each designed to analyze biological data and generate an accurate protein structure using advanced deep learning and computational techniques. First is pre-processing. So AlphaFold 2 compares the input amino acid sequence with the sequences from database to identify the similar ones, detecting correlated mutations and patterns. It also identifies the proteins with similar structures to derive the structural information. Then it has transformer module. So one transformer module processes the multiple sequence alignment data, extracting information about sequence variations and correlations. Then we have another transformer which focuses on pairwise interactions between the amino acids within the template protein structures. Then we have structure module. So the structure module treats the protein as a collection of triangles in 3D space and iteratively transforming them to form the final protein structure. Then we have invariant point attention, which incorporates three-dimensional structure information by using novel attention mechanism, which enhances the machine learning's ability. That it is like a brushing the or removing the steric clashes in a protein structure. Then finally we have self-distillation. It leverages both labeled data from PDB and unlabeled data from protein sequence repositories to enhance the predictions or to correct the errors in the prediction. Okay, so before moving here, next concept we have is protein stability. So before moving on to protein stability, let's look example of one homology modeling method, one ab initio method, and one machine learning method, that is AlphaFold. So first I'll be moving on to the Google search, then entering Swiss model, then click enter. So you will be seeing the first link, swissmodel.exposé.org. Go to the first link. So it shows Swiss model is a fully automated protein structure homology modeling server, accessible via Exposé web server. So the purpose of this web server is to make protein modeling accessible to all life sciences. So here I'm clicking on start modeling. For this demo session, I'm taking an example. So I'm filling up with the FASTA format of the sequence. So it performs a auto validation step for the input sequence. So the sequence has been validated. The email ID is optional. Then I'm running search for templates. Since homology modeling is based on the accuracy of the template, the first step is to find the templates. So it will take a couple of minutes to give the result. So I have already run these results. So for the same input, these are the top 50 templates. So these templates are obtained by performing a blast. So these are the templates that are present in the blast result along with their sequence similarity to the given template. Once templates are provided, we can use build model option here. Now the build model option is disabled because I have already built the model using this. So this is the modeled structure for the given input sequence. So when we observe on the left hand side, there is an option provided for structure assessment. along with several other metrics such as oligostate, mean deviation and all. So when we move into the structure assessment, first thing is we see Ramachandran plot and mall probability results. Okay, they'll take little time to load. And alignment of this structure with the template chain. So the template selected here is 2EFO, which shows a significantly high similarity with sequence similarity with the given sequence. Here, this mall probability score is the sequence quality score. Clash scores for the that are the van der Waals clashes or steric clashes that are experienced in this. Then we have Ramachandran favored. Number of residues according to the Ramachandran distribution are 96.93, and we have Ramachandran outliers. So we have two outliers, that is A leucine and 129 leucine at 8th residue leucine and 129 residue leucine are Ramachandran outliers, so which are not according to the Ramachandran distribution. So it also gives the information about the bad bonds, bad angles and other. So these are the parameters that are useful to assess the quality of our build model. So here the scores are everything set to 0 and scores look good because this is an example sequence. But these scores tend to change when the similarity with the template changes. So this is how, so this structure could be downloaded and can be used for different analysis purpose. Now we have ITASR. ITASR is a ab initio based method. So to access ITASR, one can search ITASR in Google. So first link is from Zhang group. So ITASR serves for protein structure and function prediction. So this is the home page of ITASR. So in this demo session, I'm not running any ITASR as it takes long time to run. So here I'm showing you an example of ITASR output which is given by them. So the first thing is what is the sequence we submitted. So this is the sequence, that example sequence. Then predicted secondary structures. So here where H stands for helix, S stands for strand and C stands for coil, along with the confidence score. Then we have solvent accessibility, predicted solvent accessibility for the structure generated. So these values ranges from 0 to 9, 0 showing for buried residues and 9 for highly exposed residues or a surface residue. Then we have a B factor values. And top 10 templates used by ITASR. So these are the templates that have been used by ITASR along with the top 5 models predicted by ITASR. So further, these models can be structurally evaluated and the appropriate model can be selected. And it also helps in prediction of ligand binding sites and cofactor, and it also gives the functional annotation, gene ontology annotations. So it gives some functional information as well along with the structure prediction. Then we have AlphaFold. So this is a Google Colab file that is available for generating the AlphaFold structures. Most of the UniProt IDs are having their AlphaFold structures available. If in case there is a structure missing, those can be modeled using this AlphaFold Google Colab. The one limitation of AlphaFold Google Colab is it cannot take input of more than 1400 residues, and the quality the quality of the structure is better when we use less than 1200 residues. More than 1200 residues, the quality is very bad when using. So this AlphaFold Google Colab is directly available. That is AlphaFold Google Colab. Search by using this as a keyword. So you'll get a collaboratory, Google Research Collaboratory. So here one can input the query sequence, and then you can give a job test and template mode, whether it should run with a template mode or a without template mode using no template information as we discussed. So it has a MSA mode which gets from the homologous sequences. It has a pair mode which uses pairwise matrix from the similar structure or template structures. Once you input your structure, default parameters can be used. Then go to the runtime, click run all. So all the cells, all the code will be run on its own, providing the output. So here is the one I have previously run here. So this is the model structure using AlphaFold. So for this AlphaFold input, I have used ITASR example input. So this is the protein structure for the ITASR input. So you can see the refined structure when compared to the ITASR input, ITASR predictor structure. Yeah. And when this code runs, automatically the file will be downloaded. And the color code, the PLDT is a quality metrics that AlphaFold uses. That is, this tells about the local deviation of the terms. So this is how AlphaFold can be used for structure generation. So the work is only to input the sequence here, then click on runtime, run all the cells, and it would take 30 minutes to 45 minutes based on the input sequence size. Sometimes it may take days based on the server load because it is running on their side, and automatically the file will be downloaded and saved in your local downloads directory. This is what demos I've done. Then we have protein stability. So protein stability refers to the tendency of protein molecule to maintain its three-dimensional structure under various environmental conditions. Or protein stability is defined as the free energy difference between folded and unfolded states. The stability of a protein structure is influenced by numerous factors, including sequence of amino acids, interactions with solvent molecules, temperature, pH, and presence of ions and other molecules. So this understanding of protein stability is essential in fields such as structural biology, biochemistry, and drug discovery. Proteins must maintain their native state to perform their biological functions effectively. Changes in protein stability can lead to protein misfolding, aggregation, or loss of function, which are associated with various diseases as well, including uh, neurodegenerative disorders like Alzheimer's, Parkinson, etc. Several methods are used to study protein stability experimentally, including spectroscopic techniques like circular dichroism, fluorescence spectroscopy, as well as computational methods based on knowledge based methods, machine learning methods, and hybrid methods are there. By studying the protein stability, researchers can gain insight into the structure function relationships of proteins and develop strategies to modulate protein stability for therapeutic process. So in general, protein stability is defined as a Gibbs free energy of unfolding in absence of denaturing concentration. So we'll see how it is defined. So just a brief introduction to Gibbs free energy equation. It also known as Gibbs-Helmholtz equation, which relates as the change in Gibbs free energy to enthalpy change and entropy change and temperature. That is, delta G is given as change in enthalpy minus temperature into change in entropy. So in the context of protein stability, a negative delta G indicates protein thermodynamic is favorable, that is stable, whereas delta G positive indicates that unfolding is favored. So protein stability is determined by the complex interplay of various factors, including protein structure. The folding of protein into its native conformation is a result of delicate balance between the attractive forces and repulsive forces among the amino acid residues. The structure of a protein is stabilized by a variety of interactions such as hydrogen bonds, disulfide bonds, van der Waals interactions, and hydrophobic interactions. The forces which drive the protein folding are the hydrophobic effect, hydrogen bonds, and van der Waals interactions. Yeah, X-ray structures can be used. There are several statistical equations that are available. Foldex is one such uh, tool that is available, which can calculate overall folding energy and its contributions from enthalpic terms, entropic terms, water terms, and and it also gives. So for more detailed information, Foldex gives it gives overall folding energy, followed by that what are the different contributions, enthalpic contributions and enthalpic contributions to that folding energy. So here the forces that promote folding are non-covalent interactions mainly and hydrophobic effect, but the force which oppose folding is chain conformational entropy. So the balance between those opposing and supporting forces is the net protein stability. And the solvent environment, including pH and temperature, can affect the stability of a protein by altering balance between electrostatic and hydrophobic interactions. So to study about individual contributions of these to protein stability, the hydrophobic effect plays a crucial role in stabilizing the protein native structure. When non-polar amino acids are buried in the hydrophobic core of a protein, they minimize their exposure to the surrounding aqueous environment. This minimization of hydrophobic surface area reduces the unfavorable interactions between non-polar residues and water molecules, thereby contributing to stability. So in an unfolded state, when these hydrophobic molecules are exposed to water, the surrounding water entropy is reduced. The water becomes like the dynamic nature of the water loses. They forms a cage-like structure around these non-polar residues. During folding, these non-polar residues are buried inside the protein structure, reducing their exposure to the surrounding water. Water. So thereby, even the system is losing its entropy, but the surrounding is gaining its entropy. This is how hydrophobic effect plays. Uh, in protein stability. Then we have van der Waals forces, which include both attractive and repulsive forces between atoms and can significantly contribute to the protein stability. These forces arise from fluctuations in electron density within the molecules, leading to the dipoles or induced dipoles. So van der Waals interactions are primarily enthalpic in nature, and the attractive forces between atoms in close proximity result in favorable interactions, leading to the decrease in enthalpy and contributing to the stability. Then we have hydrogen bonds. We know they are the stabilizing the stabilizer for structural motifs on secondary structures. Then we have chain conformational entropy. The folding of a protein involves decreasing conformational entropy of a polypeptide chain or a protein as it transforms from highly populated denaturation state to a unique folded state. Entropy is nothing but amount of orderness or disorderliness in a system. That's a measure. So this decrease in entropy acts as a barrier to the folding and opposes the stabilization of the native protein structure. Despite this entropy loss, the favorable contributions from non-covalent interactions outweigh the entropic cost in most of the native state proteins, resulting in a small negative delta G favoring protein folding. So what is the advantage of this delta G being very small? That is, this could be easily altered by the point mutations so that the protein effects can be removed or protein effects can be enhanced. So understanding the interplay between these factors is essential for elucidating the thermodynamics of protein folding and stability. Experimental studies, including analysis of wild and mutants, provide valuable insights into the relative contributions of each factor to the overall stability of proteins. So recently, we have uh, published the first work on transmembrane alpha helical protein stability, and we also find out what are the factors contributing to the alpha helical transmembrane protein stability in their membrane environment. Do check out if you have some time. So by investigating all these thermodynamics of small molecule interactions and behavior of proteins under different denaturation conditions, we can dissect the mechanism underlying the protein stability and folding. So I have few set of questions, and then today we'll be trying to solve mostly the center of mass based questions. The first one, which of the following structure prediction method does not rely on a template structure? Yeah, so here the option D, integrative modeling, defines the combination of homology or ab initio or threading or ab initio, like that. Integrative means integration of one or more methods. So which does not rely on a template structure. Even the template, even recognizing fold recognition is part of template only. So here the answer is B, that is ab initio methods. Homology modeling is completely based on template. Threading identifies the similar folds that are present and then try to build on those folds. Integrative modeling is again, since combination of other methods, it's again a template based method. So we have ab initio method, which uses the physics based or quantum based properties and develop a huge collection of conformations and then try to identify the conformation with least energy and defines it as a native state conformation. Then which of the following is dominant in unfolded state of a protein? Whether hydrophobic interactions, van der Waals interactions, electrostatic interactions, or entropy. Which of the following is dominant in unfolded state of a protein? So entropy in unfolded state is highly dominated by the entropy of the system, whereas the non-covalent and the interactions such as hydrophobic, van der Waals, and electrostatic dominate in folded state of a protein. So which of the following interactions is modeled using Lennard potential during MD simulation? So which of the following interactions? So Lennard potential, whether it is used for modeling van der Waal interactions, electrostatic, or both, or none of the above. Okay, I know this is a little out of context question. Yeah, it is used for van der Waals interaction. This comes at, thanks for your answer. This is little kind of out of context question. Yeah. So which of the following methods is used for measuring folding free energy or for knowing the stability value of a protein? Folding free energy, delta G. Which of the following method is used for measuring folding free energy? So all these experimental techniques employ either thermal denaturation or chemical denaturation studies to measure the effect of denaturant concentration on folding free energy. This folding free energy value, we call it stability whenever we take it at zero denaturant concentration. For example, remember a plot. At one denaturant concentration, you have a delta G value 1. At 2 denaturant concentration, you have delta G value 2. Similarly, you have n delta G values for n different concentrations. When you extrapolate this plot to the place where the x-axis value is 0, the corresponding y-axis value will be the stability. That is how stability value is defined. There is no direct measurement for protein stability values as such. Now, I'll be showing in a couple of minutes how this plotting is done. This is just for so differential scanning calorimetry is mainly used for knowing the melting temperature and melting thermal stability. So here all of these methods are used for measuring the folding free energy or stability of a protein. So here now we have a true or false question. Rosetta is a web server that predicts the protein three-dimensional conformation using the ab initio method. Is Rosetta a ab initio method? True or false? So Rosetta is not a template based method. So since it is not a template based method, whether it could be ab initio or not. If it is ab initio, respond true. If it is not, false. Okay, no one wants to answer. Rosetta is ab initio based method since it doesn't take into account any homologous template structure while structure prediction. So which of the following is used for validating model protein structures? ProCheck. ProCheck is used for validating model protein structure. This ProCheck is also part of Swiss model, but a demo we have seen. Yeah, so this is a kind of a thinking question. So you have a protein structure and you want to quickly predict its structure. After performing blast searches, you identify that most closely related proteins with known structures having 15% amino acid identity to your protein with a non-significant expect E value. So which of these following options will be correct to predict the structure? Like you want to predict quickly, but yeah, you have a template with only 50% amino acid identity, that too with a non-significant expect value. So I'm repeating the question here. Here we want to predict the protein structure quickly, and in the blast we have observed the template is having only 50% amino acid identity. That identity is also, the probability is low. The probability that this identity could be wrong is high. Then we'll tell non-significant expect value. So what of the following could be done? So this non-significant expect value. So first, expect value is a parameter that describes number of hit one can expect to see by chance when searching a database of particular size. So it is nothing but e-value describes a random background noise. The smaller e-value, the better match, and probability that our alignment match is correct is high. It's a e-value. Non-significant means it's high e-value. Telling that even this 50% amino acid identity is by chance. Yeah, actually, when it comes to the cutoff, e value having e to the power of minus 50 is, we tell it's a very high quality or very good e value. But when we are doing this homology matches and all, 0.01 could be a still be a good value. Like approximately to tell 10 to the power of e to the power of minus 50 is very good value to get. And for homology matches and all, 0.01 e value or anything less than 0.01 could be good. So anything other than that is termed to be a non-significant. So any answers to this seventh one? Yeah, good alignment. It represents the good alignment. Smaller the E value, the better is the alignment. So here moving, so that since we don't have a good template structure that is readily available for homology modeling, so option C we couldn't perform. Then extra methods and NMR methods are time taking, so quickly the question asks quickly to predict. So we have left with ab initio modeling methods which can give the protein structure when there is no significant homolog is present. So here I have given the coordinates, XYZ coordinates of a PDB 1BTA, and particularly this coordinate represents for alanine 14. So using these coordinates, try to calculate the center of mass. The formula for center of mass for X coordinate has been given. Similarly, it should be calculated for Y coordinate. So the atomic masses of nitrogen could be considered as 14, carbon could be considered as 12, oxygen has 16, hydrogen has 1. So the general chemistry atomic masses could be considered. Nitrogen has 14, carbon has 12, oxygen has 16, and hydrogen has 1. So to start with the calculation, consider the X coordinates. The first is for atom N. So atomic mass of nitrogen multiplied by its X coordinate plus atomic mass of carbon C alpha, that is carbon atom, multiplied by its X coordinate plus atomic mass of carbon multiplied by its X coordinate, then uh, atomic mass of oxygen multiplied by its X coordinate, and atomic mass of carbon multiplied by its X coordinate. Sum of all these values divided by sum of masses of N, C alpha, C, O, and C beta. Okay, I have two more similar questions. Let's see solution of this. So here for X coordinate, have considered first N atom. Atomic mass is 14. 14 multiplied by its x coordinate. Then C alpha atom atomic mass is 12 multiplied by its x coordinate. Next again C atom atomic mass is 12 multiplied by its x coordinate plus oxygen atom its atomic mass 16 multiplied by its x coordinate. Again C beta atom its atomic mass 12 multiplied by its x coordinate divided by sum of the atomic masses of all the atoms, that is N, C alpha, C, O and C beta. Similarly for Y, instead of x coordinate here, replace them with the Y coordinates. You will get center of mass coordinate of Y and Z. This is how a calculations are performed. So I'll wait here. Please glance through the calculations. Don't worry about the final value. That's just division. I have two more. One we can solve, and then we can go to take on some stability calculations. Okay, so here is the similar second question for calculating center of mass of glycine as in given coordinates here. So the similar atomic masses can be considered: nitrogen 14, carbon 12, oxygen 16 and hydrogen 1. So do try to solve these calculation problems, because in exam it helps you in faster calculation. The number of questions you practice, the number of pro you become in these, and it would be easy for you to solve these kind of numericals in exam, and it also helps in a time management in the exam. Yeah, these are basic physics formula and simple mathematics only, but many of them does mistakes in these cases. So a consistent practice is needed. So why I have given three questions here. Previous years we have noticed many people doing wrong in these calculations, writing equations correctly and messing up with the answers. In exam, will they give some answer center mass choices to select? Now, kindly confirm with the NPTEL team. I that exam center details and all we are not
Uh,
getting it.
Generally, I have seen for such questions, they give center of mass A, B, C, D, and they give the X, Y, Z coordinate. One of them we have to select.
Oh, this is
That's why
center
I
of
asked.
mass.
Center
Okay,
of
sorry,
mass.
sorry. I mistook it for center, for exam center and all. Center of mass will, if we are asking, we'll be giving the PDB format and ask you to calculate the center of mass. And
And they will be giving the answers like A, B, C, D, which one to select. That's what I have seen. Will that be that format, or they will ask the center of mass to be collected and entered?
yeah, see, actually, NPTEL exam has both type of questions. It could take numerical, it could take a descriptive one or two line, as well as it is an MCQ.
Okay,
So,
thank you.
yeah. Okay, I'll send this. I try to solve these two by your own demo sessions we have done. So we have some quick few questions on protein stability. With that, we can close this. So what is the primary driving force for protein stability or protein folding? Out of all the interactions, what is the primary driving force? So how can one tell which is a driving force? So based on its contribution, like for example, when a protein we observe, what is the number of electrostatic interactions present? To the residues contributing ratio normalized to the number of what I tell residues contributing to electrostatic interaction. Hydrophobic effect. Number of residues that are involved in hydrophobic effect. Number of hydrophobic interactions divided by number of residues that are involved in forming hydrophobic interactions. Covalent bonds. Number of covalent bonds divided by number of residues that could probably involved in covalent bonds. Similarly, van der Waals. So higher the value we tell, they are the primary driving forces. These normalized values. So here hydrophobic effect is the major driving force, not van der Waals and other things. Because hydrophobic collapse is the major driving force because it would help in the collapse of hydrophobic residues in the interior of the protein as well as the surrounding water, reducing the orderedness of the surrounding water. So this way hydrophobic effect is the major driving force. So which interactions stabilize the secondary structure elements of proteins? Which of the following interactions stabilize the secondary structure elements of proteins? Whether they are hydrogen bonds, van der Waals forces, disulfide bonds. Secondary structure elements. So what types of bonds generally we see in secondary structure elements? If you know that, it would be easy to answer this question. The hydrogen bonds. So the answer is hydrogen bonds. So which of the following can destabilize a protein structure? As we have discussed, we have effect of the protein stability depends on interaction of it with other residues, interaction with the environment, and physiological conditions as well. So which of the following can destabilize protein structure? Yeah, higher temperature. Yeah, any temperature above than its uh, optimal temperature could change its folding pattern and thereby leading it to loss of function or cause of disease or cause of aggregation. So given a kind of a thinking question, at what temperature would you expect protein denaturation to occur more rapidly? Consider room temperature is the optimal temperature for this protein. At what temperature would you expect protein denaturation to occur more rapidly? Considering this protein is having a functionally optimal room temperature. Yeah, it's 100. So more the temperature, more the denaturation to happen quickly. This is kind of some a thermodynamic based question. Calculate the change of entropy if protein unfolds and its conformational entropy increases from 200 kilojoule mole Kelvin to 300 joule per mole Kelvin. So we have seen entropy increases from 200 to 300. So initially it is 200 and it has been increased as the protein unfolds to 300. Yeah. So here it is initial entropy is 200 and final entropy is 300. So 300 minus 200 is the entropy change if protein is unfolding. Yeah. So what is the change in Gibbs free energy of a protein folding process if enthalpy change delta H is minus 300 kilojoule per mole and change in entropy delta S is 150 joule mole per Kelvin and temperature is 298 Kelvin. Consider here the units of enthalpy and entropy are not same, like they are same but not in the same order. So kindly have a look into this before solving. Let's move on to this solution. So here the given enthalpy value is delta H is minus 300 which unit kilojoule per mole. Here we have units of Kelvin and 150 joule per mole and Kelvin. Since to convert this joule to kilojoule, I am just subtracting it by dividing it by 1000. So this gives minus 300 minus 288 into 150 divided by 1000. This gives minus 300 minus 44.7, which equals to minus 344.7 kilojoule per mole. Yeah, so these are the questions that I have for today. Thank you for attending the session. If you have any doubts, please ask me. If not, you are free to leave.